Welcome everybody. I hope you had a nice weekend. Today we have Dr. Pividori speaking. Dr. Pividori is an assistant professor at the Department of Biomedical Informatics at the University of Colorado. His lab aims to design the next generation of computational methods that consolidate large and heterogeneous sources of biomedical data to extract biological insights to ultimately improve human health. So I'll leave it to you now, Dr. Pividori. Thank you very much.
Fantastic. Thank you so much. Thank you, everyone. Thank you for this invitation for joining this presentation today. So yeah, I'll be talking about genetic studies through the lens of gene networks today. First, I wanted to talk a little bit about myself. I'm originally from Argentina. In Argentina, we drink mate. It's a hot beverage, very popular in South America. If you like soccer, probably you heard about Maradona. He was one of the best soccer players in the history. And if you follow the MLS, Messi is playing in the Inter Miami. So I'm an average soccer player, not very good, but I really enjoy watching it. So regarding my research journey, I got my PhD in Argentina. I'm a software engineer by training. My PhD was on computer science applied to bioinformatics. Then I went to the University of Chicago, where I did a postdoc. I studied genetics. Then I continued my journey of software methods development by incorporating omics data integration pipelines. And that's how I became a biomedical informatician. And as Ahmed said, I'm an assistant professor at the University of Colorado Anschutz Medical Campus. So in our lab, our research is focused on developing and applying advanced machine learning methods for knowledge discovery, hopefully enabling a multi-omics approach to precision medicine by integrating heterogeneous, diverse data modalities. So today, I wanted to very briefly introduce genome-wide and transcription-wide association studies, how we apply those methods to a particular disease, asthma. And then talking about networks, integration with genetic studies, how we use this method for different purposes, and a little bit of the ongoing and future research in the lab. So I'm sure you heard about this concept a lot. Precision medicine is defined as an emerging approach for disease treatment and prevention that takes into account individual variability in genes, environment, and lifestyle of each person. This presentation and what we do in the lab is to focus on the genetic component of this variability. So in genetic studies, our goal is to understand how this genetic variation leads to phenotypic variation. For example, we want to know which genes are linked with particular diseases. And genetic studies are important because they can contribute to two broad goals of precision medicine. The first one is to elucidate, understand the biological mechanisms and identify potential therapeutic targets for a particular disease, and also to predict individuals at risk of disease, for example, and make changes in the lifestyle, for instance. So a very well-known approach are genome-wide association studies. If we think about tables and data frames, what we have here is the genotype matrix, where we have SNPs across thousands of individuals, and we have a trait of interest, which could be a continuous trait or a binary trait in case of a disease, for instance. So what we do is to take every single nucleotide polymorphism in the genome for which we have measurements, the trait of interest, and we compute a simple regression model, which could get more complicated with newer approaches, and measure the effect of these SNPs in this trait. So the output of a GWAS mainly is the effect size or the slope of the linear regression model, and the p-value representing the significance of this association. So usually when you want to convey the result of a GWAS, you show a plot like this Manhattan plot, where you can see each point is a SNP. In the x-axis, we have the position of this SNP in the genome, and the y-axis shows the significance of that SNP. So you can see in this example, different regions in the genome that are significantly associated with a particular trait. However, GWAS alone does not give us a mechanistic understanding of these associations. What we do in a GWAS is to compute an association between these SNPs with the observed phenotype, but we don't know what's going on in the middle, how those variants are affecting different molecular mechanisms, phenotypes, for example. There is a bunch of different methods to test that, like transcriptome-wide association study, which tests the effect of these variants on expression, or protein-wide association studies, or those who incorporate different molecular mechanisms like methylation, etc. I'm going to focus today on TWAS, which is the method that I have experience with. So here we map these variants to genes. What TWAS does is to build prediction models of gene expression. So we need genotype and expression, and we predict expression from genotype. And then we can use these models to compute a gene trait association or predict expression in a cohort where we don't have expression, like the UK Biobank, for example. So once we predict expression, so this here we have the same matrix, same individuals, but we have the predicted expression of these P genes for a particular tissue. We take, again, our trait of interest. We use, again, a linear regression model to compute an association between each of the genes with the trait of interest. So what TWAS does is to test whether these variants have an impact in gene expression, and that expression is mediating, actually, the association with these traits. So let me show you how we use these two approaches in a particular disease of interest. This was when I was in Chicago. We studied asthma. Asthma is a chronic respiratory condition that affects the airways in the lung with clinical symptoms such as wheeze, cough, shortness of breath. However, when we say asthma, we actually refer to an umbrella term that comprises different syndromes, which could also overlap. For example, at the age of asthma onset is a key discriminating factor in identifying these different subtypes or asthma phenotypes. Two of them are childhood onset and adult onset asthma, which differ in many aspects. For example, adult onset asthma is more common in women than in men. Prognosis is usually worse, it's less allergic. It has a poorer response to therapy. So our question in this study was whether this phenotypic heterogeneity was reflecting differences in genetic risk factors and molecular mechanisms. So for this, we used the data from the UK Biobank. We defined these groups with the sample size that you see here, especially the childhood onset asthma, adult onset asthma, and the controls. You can see that we have a larger sample size for adults. And these are the results. This is a Manhattan plot for the childhood onset asthma. Again, in the x-axis, you have the position of the SNPs across different chromosomes. The y-axis shows the significance of the association of these SNPs with childhood onset asthma. And you can see different peaks that are associated with this asthma phenotype. And in this plot below, you can see the results for adult onset asthma in red. This is what we call usually a Miami plot. You can see that we have fewer significant loci for adult onset asthma, even though we had almost 2.5 times more sample size for this group, which was striking for us at the beginning. So this kind of analysis, for example, showed us that there are regions that are specific, in this case, to childhood onset asthma. This is a very well known and replicated region in chromosome 17 for childhood onset asthma. You can see also that there are most of the regions are childhood onset specific. We also found in chromosome 2 the only adult onset specific locus. And the analysis also highlighted shared regions like this HLA region that had two independent associations. So this was, in the time we performed this study, was the largest GWAS of these two asthma phenotypes. So in general, you can see that the genetic risk factors of adult onset asthma seem to be a subset of those of childhood onset asthma, where we have fewer significant loci, generally with a smaller effect sizes. And this suggested that there is a greater role of environmental risk factors in adult onset asthma. So this is, I'm describing this very quickly. This is not the main point of the presentation, but it's a necessary introduction. So here we perform this single variant-based analysis. We also did the single gene-based analysis, that is TWAS. Here are some results. We focus on 5 tissues of interest for this trait. So here in this plot on the left, each point is a gene colored by their position, their chromosome. The x-axis has the significance of the association measured as a Z-score for childhood onset asthma. And the y-axis is the same, but for adult onset asthma. And you have different dashed lines that represent different thresholds of significance. So all these genes here are childhood onset specific, as you can see. So for example, if we take PGAP3, which is a gene in chromosome 17, you can see that it has a positive Z-score, which means that a higher predicted expression of the gene is associated with asthma risk, in this case, childhood onset asthma. However, this other gene on the left, GSDMA, is also a gene in chromosome, the same region, chromosome 17, but has a negative Z-score, meaning that a lower predicted expression of this gene is associated with asthma risk. So you can see that we have different patterns of association with positive, negative Z-scores. I think it reflects the broad and complex regulatory effects of these SNPs in this loci. Here on the right, I'm showing some genes that were shared across these 2 phenotypes in the skin, but they were generally more significant and with higher effect sizes in childhood onset asthma. So that was the single gene approach. The third analysis that we did here was to create 2 groups of genes, those associated with childhood onset asthma and those associated with adult onset asthma. And we perform a tissue enrichment analysis to gain an even higher understanding of the molecular mechanisms in asthma in this case. Something that I didn't mention is that if you have any question, please interrupt me at any point. Sorry, I forgot to mention that at the beginning. So here are the results. The tissues that you see in the x-axis are from GTEx. So here we can see that genes associated with both childhood onset and adult onset asthma were more highly expressed in whole blood and small intestine, highlighting a shared immune cause for these 2 asthma phenotypes. The genes in childhood onset asthma were more highly expressed in the skin, which I think it shows that this could be a primary tissue, target tissue for early onset asthma. And those associated with adult onset asthma had an enrichment in the lung, so suggesting a more lung-centered cause for this group. So this was the first, as a summary, the first large-scale genetic study to provide evidence for a greater genetic component in early onset asthma. It suggested shared immune mechanisms, but also distinct mechanisms. In particular, we suggested that the establishment of disease in children is driven more by dysregulated allergy and epithelial barrier function genes, whereas the cause of adult onset asthma is more lung-centered, and as I showed you in the GWAS results, potentially more environmentally determined. So the conclusion in general is that more effective treatments might differ between these 2 groups, and the strategies, if we want to implement a precision medicine approach, should be potentially further personalized to account for the age at asthma onset. So in this study, we applied a single variant approach, a single gene approach, and we created these 2 very basic groups of genes and performed some tissue enrichment analysis. However, another step is to make this group of genes more sophisticated, or to capture groups or networks of functional related genes. So if we talk about networks, gene regulatory networks in the context of genetic studies, we have to cite or talk about the omnigenic model for complex traits. This is an idea, a theory that was published in Cell in 2017. And it is important because most of the GWAS variants are in the non-coding region, more than 90% of them. Their effect sizes, both in GWAS and TWAS, are usually modest or small. That means that they explain a small portion of the trait variants. And this limits our ability to understand the genetic basis of different traits. So what the omnigenic model basically says is that gene regulatory networks are so highly interconnected that potentially all genes expressed in a disease-relevant cell might have a direct or indirect effect on the trait. So in particular, this model defines two categories of genes. They define core genes, which are these black stars in the plot that you can see here, which are thought to have a direct effect on the phenotype without mediated regulation of other genes. So the product of the gene here directly affects some trait of interest, for example, the levels of lipids. And the peripheral genes, which are the gray circles, which indirectly affect the phenotype by regulating these core genes. So, of course, this is a very likely a simplification, oversimplification of the biology. They are, for example, mentioning just a binary classification of genes. However, some genes might be more peripheral than others. Or maybe some diseases may not even have core genes. Maybe the global activity of genes might help to set the state of the cell to determine its function or disease risk, which was suggested by other results. But I personally like this model a lot because it offers, I believe, a conceptual framework to start thinking about our results in genetic studies with an explicit hypothesis of the genetic architecture of complex traits. And importantly, these core genes might represent more, if they exist, might represent more attractive candidate drug targets. So how can we extend our approaches to incorporate these networks? So one approach is to use gene co-expression networks. Sorry, I think Chris has a question.
Yeah, I have a question, a brief question. Can you hear me?
Yes, I can hear you.
Okay. My question is that sometimes in these gene networks, we have these genes that are like I guess ma what we call master regulators, where a single gene affects many downstream targets related to phenotype. Under this model, would you consider those to be core genes or peripheral genes, since they're often thought to have such a profound impact in many aspects of phenotype?
Yeah, that's a great question. I don't think the model provides a lot of details on that. Yeah, my take is that I would consider it probably as a peripheral gene, but as I said, it might be another category of peripheral gene. Maybe it's closer to a core gene type of function, especially if you, I will show some results later, but maybe if you knock out the gene, it has a huge cascading effect on the phenotype. But the definition, I mean, if we take the pure definition from the omnigenic model, is that the core genes do not regulate other genes. Their product directly affect the phenotype. So that's the definition. But then we don't know. I mean, we could have other types of definitions, like the one, for example, that you suggested. There is a paper from 2019 where the same authors propose a method that incorporates these ideas of master regulators, for example. Am I answering more or less your question?
Yeah, I think so. Thank you very much.
Sure, we can go deeper later if you want. So one idea here is to incorporate these ideas of networks to use large RNA-seq data where we have measurements across different conditions. For example, here we have measurements on macrophages, for instance, under a viral infection. You can see that genes A and D in particular are highly expressed. And here we have another example where a different set of genes is expressed in the heart. So the idea is to apply some, for example, unsupervised method to find these modules, gene modules, which are defined as group of genes that have a similar expression profile across the same cell types. So this is not included specifically in the Omnigeny model, but this is more or less what we think about this biology. So these modules are expressed in a specific context, in specific cell types. They could have a cascading effect if they are peripheral genes, if they are master regulators. They could, as Chris said, they could have huge effects on different genes. But the idea, we think also about this modular idea of genes. You can see how modules could even share genes also. So the way we obtain these gene co-expression models is by applying, for example, Plier, which is a matrix factorization approach for expression data. So methods like Plier give us basically two outputs. The first output is how these genes are clustered together in these modules. In this example, you can see how genes A and C have a high weight in gene module 1, meaning that they belong to this gene module, whereas gene A and D have a high weight for gene module 2. So you can see that these genes can participate in different gene modules. Another output that we can get from Plier is sample weights for each module. So for example, here the output is telling us that genes in module 1 are grouped together because they are expressed similarly in the heart. And in gene module 2, they are expressed similarly in macrophages. So the idea is to integrate these modules where we have groups of genes and where some indication of where they are expressed with TWAS, where we have associations across thousands of different traits. And the goal is to connect these modules with a particular trait of interest. For example, gene module 1 might be expressed in the heart and associated with a cardiovascular trait. And gene module 2 is another mechanism expressed in immune cell types and also associated with the same trait in this case. So we call this approach PhenoPlier. And we evaluated or developed different methods to test this idea of gene modules. The first approach is a regression model that can compute an association between a module and a trait. And here we work at the gene level, not at the individual level, as GWAS or TWAS. And the basic idea is to predict the trait associations across genes. This is our Y, using the gene weights for a module. So basically, the hypothesis we are testing here is that genes with high weights that strongly belong to the module tend to be more strongly associated with the trait of interest. And of course, we need to take into account in this model gene-gene correlations, since, for example, in TWAS, we could have EQTLs or single SNPs that could affect the same gene or be NLD. So the TWAS statistics could be correlated. So the output is an association, as you can see here, a p-value of between a module and a trait, as you can see here, for instance. So that was the first computational development. The second one the idea was to learn shared and distinct transcriptomic processes across traits. So we use this latent space that we learned from expression data, and we projected these TWAS statistics into this space, and then ran a clustering approach to group traits together and try to learn shared and distinct processes across these traits. I will give you more details later. And then we also used drug repurposing framework that was previously published. So what we did here was to use drug-induced transcriptional profiles that were also projected into this space. So in addition to the TWAS associations that brings traits into this space, we also projected drug disease associations and perform a joint analysis. And I will show you more about this later. So the first aspect that we evaluated is the prioritization of candidate therapeutic targets. When we did this study, it was at Penn. The team that worked on this project was interested in lipid metabolism. So one of the labs performed a fluorescent-based CRISPR screen with the goal of identifying genes strongly linked to lipid accumulation. So in the image, you can see two groups of cells. The one in the middle, in panel B, are those with a viral transduction where a fluorescent dye was diluted to stain lipid droplets. So this process identified 462 genes associated with lipid accumulation. From those, we identified a high confidence lipid increase in gene set with these 6 genes, in particular these ACACA and DGAT2 that I will mention later. And we took this gene set and performed gene set enrichment analysis across all our 987 modules that we had. And we identified 15 nominally enriched modules. And this is the top module that was nominally enriched for these 2 genes. We identify it as LV246. When we look at where, in which context this module was found to be expressed, we found that it had a strong adipose tissue signature. Here, each point is an RNA-seq sample projected in this space. And the y-axis is the expression of those samples in this LV, and the x-axis are the tissue cell types. So you can see that it's very adipose tissue related with some other interesting tissues like liver, astrocytes. When we use our regression model to compute a module trait association using the phenome scan, which is basically a set of results of TWAS from the UK Biobank, we found that the associations were highly lipid specific, as triglycerides, LDL cholesterol, or conditions like high cholesterol and even Alzheimer's and dementia. We replicated most of these associations in eMERGE, which is another cohort. We found this LV associated with hypercholesterolemia, also memory loss. Something else that we did here was so we found all these associations at the module level, but we wanted to look at the individual TWAS associations. So what was going on at the individual level? So we took the top genes of this LV246. These are sorted by sample weight. So SCD is the gene with the largest weight here. And then these are the traits associated found by the module, our regression model. And here, something that was very interesting for us is that these 2 genes, DGAT2, ACACA, that were found in the CRISPR screen have very large p-values. So they are not significant with any of these traits that were found to be significant at the module level. Actually, they are not very significant across any of the traits. So we wonder why this was happening. Our hypothesis is that, again, if we bring this omnigenic point of view is that maybe the genes, the other genes in the module are acting potentially as peripheral genes. And these DGAT2 and ACACA are what we can define as core genes here. And that is because, or in other words, our gene prediction models from TWAS, we have SNPs that act in cis to regulate the expression of these peripheral genes. And that's actually the only component of expression that we can capture with TWAS. So maybe this gene module approach is inferring or capturing these trans effects. That could be a potential hypothesis that we are working on. One of the students in the lab is working on to check that. So these results basically suggest that we can improve potentially the prioritization of candidate targets. Also importantly, provide a more mechanistic understanding of these associations. So that's in terms of finding potential important genes. The other part was to test this approach to learn, share, and distinct transcriptomic process across traits. So here we assume that a module is capturing some of these processes, and we want to understand whether different groups of diseases are affected by the same processes. So what we did here is to take the input data, the TWAS associations that we call PhenomeScan, which is basically based on the UK Biobank, where we have traits in the rows and these almost 1,000 latent variables, perform some clustering analysis. So for that, we generated some diversity from the point of view of the data. We projected this data with PCA and UMAP. Then we used a bunch of different clustering algorithms with completely different assumptions on the data structure. For example, k-means can only find spherical clusters. Spectral clustering is more complex. DBScan can find density-based clusters, etc. So this is a consensus clustering approach where we generated an ensemble. So traits are now grouped into these different partitions. One partition comes from different algorithms or a different set of parameters for one algorithm. And using this ensemble, we derive a co-association matrix. A co-association matrix is basically a new similarity matrix based on the clustering solutions. And then we apply the consensus function at the end to find this group of traits. And something that we wanted to do here is to understand which LVs were driving these clusters. And since this is a very highly nonlinear transformation of the data, we trained decision tree classifier using these cluster labels and the input data, and found the top LVs that were helpful to distinguish these clusters. And then we used the emerge, our replication cohort, to understand or assess those top LVs. So this is a subset of the data. Here you can see traits on the columns with color by cluster. Then we have LVs that are helpful to distinguish at least one of these clusters. And you can see different set of trait clusters, for example, these red blood cells. And we found that the LVs that help discriminate this cluster were expressed in different early stages of erythropoiesis, which made sense. And when we look at the module trait associations, they were associated with different assays on red blood cells. Here we have another cluster with platelets. We observed that the LVs that help discriminate this cluster were expressed in thrombocytes or platelets. This is from cancer samples. And this LV was also associated with platelets from different traits from the UK Biobank. So this suggests that this approach could facilitate the identification of these shared and also distinct mechanisms between different diseases. And another angle, as I said, that we use to assess this gene module perspective in genetic studies is to build this interpretable drug repurposing framework. So for that, we used a framework that was published before and applied to different in the psychiatry field. So the basic idea of this framework is that we compute a drug disease score by anti-correlating the gene trait associations and the drug disease associations. So here, D are our drug disease scores, M are our gene trait associations, and L are the drug gene associations from some drug-induced transcriptional profiles database like links L1000, for instance. That's the one that we use. So this is an example of a positive association between a drug and a disease. You can see that, for example, in this case, gene 1 has a positive C-score. That means that a higher predicted expression from TWAS, this comes from TWAS, a higher predicted expression of the gene is associated with the disease. And the C-score from links L1000 is negative, meaning that this particular drug lowers the expression of gene 1. So you can see that if we match these reverse transcriptome patterns, we can accumulate evidence of a potential association between this drug and this disease. So what we did was to apply exactly the same framework, but at the gene module level instead of genes, which had the advantage of being interpretable features. So here we can guess where we can infer where these modules are expressed. So we used a manually curated gold standard set of drug disease medical indications for more than 300 drugs and 53 diseases to evaluate the prediction performance. And here you can see the ROC curve at the left and the precision recall curve on the right. Strikingly for us, we did not perform worse in predicting drug disease pairs, but even slightly better. And I said strikingly because what we were doing here was to go from this data where we have associations for different traits across 22,000 genes, and we are projecting this data into this lower dimensional express. So this implies necessarily some information loss unless we are capturing some biologically meaningful patterns, which seems to be the case for this particular application. Then we analyzed niacin. One of the co-authors pushed a lot in adding some controversy to the paper, so we picked niacin, which was indicated for atherosclerosis and coronary artery disease in the gold standard set that we used, built in 2016. That's an important year.
Hi, Dr. Milton.
Yeah, go ahead.
We have a question from Varsha.
Oh,
just to let
sure.
you know, I mean, feel free to take it.
Yeah, thank you. Thank you. Yes, go ahead, Varsha, please.
Hi, sorry, I didn't mean to interrupt or anything, but I just had a question on the previous, I think it might have been before you brought up the drug trait association when you were discussing the hierarchical clustering.
Oh, sure.
I'm curious because I found this very interesting, of like this almost reverse engineering of diseases where you're going from traits first. And I see how like at the bottom, you have these cardiovascular diseases, respiratory diseases, and I could see how that would work for diseases that are pretty clear cut. Have you run into any issues with diseases that have slightly fuzzy boundaries, like lupus or autism or something like that, where it affects multiple different ranges of functions within the body, and it ranges from person to person, how it expresses itself? And how would you deal with that in hierarchical clustering?
Yes, that's a great question. Just in case this is not hierarchical clustering, what I was showing here is the decision tree. So what the decision tree is doing is to take some label. For example, we found all these traits to be grouped together. We built a binary classification problem, and then we use a classifier on a test set to capture which of these LVs are being helpful for the classifier to distinguish these traits. So basically, why these traits were clustered together. So this was one of the analyses that we performed. Your question is great. We haven't delved much into that. We found that I think I showed it here. We found that the top LVs that were helpful to distinguish in this case, where is this? Some simple red blood cells made sense from the point of view of where they are expressed and which traits were associated in the discovery and replication trait. But that would be that's one of the goals what you mentioned. When I was gonna talk about niacin, I was gonna mention two mechanisms of action that are pretty different. So yeah, we potentially could find modules expressed in different tissues resembling potentially different functions. So that's the goal of this module approach. We didn't delve much into specific diseases and coming up with lists of modules that represent different functions, as you suggested, that could explain these different, molecular mechanisms that could be affecting a disease. But that's a potential goal and a very interesting analysis.
Thank you. Thank you for explaining.
Sure. So maybe the next example says something about what you just asked. So we took niacin again. So it was indicated for atherosclerosis and coronary artery disease in this gold standard set that we used, and was predicted as a disease-modifying indication for cardiovascular diseases in our framework. Of course, we have a huge however here. We need to mention that niacin as a monotherapy was associated with a reduction of some cardiovascular events into all clinical trials, but we have more recent clinical trials data, and they do not suggest niacin as to prevent cardiovascular disease, mainly because there has been a substantial advancement in cardiovascular disease management. So maybe the aggregate effects of these interventions change the underlying risk of cardiovascular events, and that reduce the marginal effect of niacin. But here our framework does not capture interactions between drugs. So I wanted to mention that. So what we did to try to understand why our framework was making this prediction is to take the score between niacin and different cardiovascular diseases and decompose it. So, for example, we found that LV246, this module that I talked about before, very related to lipid accumulation, was among the top 20 modules contributing to the niacin prediction of several cardiovascular diseases. And this was interesting because this module is expressed in adipose tissue aligned with lipid metabolism pathways. And this was resembling one mechanism of action of niacin that decreases LDL production by inhibiting adipocyte triglyceride lipolysis. And we found another module, LV116, expressed in macrophages and different immune cell types aligned with immune-related pathways. And this was resembling another mechanism of action that was found later for niacin that inhibits atherosclerosis progression in immune cells. This could be potentially found also not at the drug repurposing framework, but if you are analyzing clusters of diseases, maybe you can find different LVs resembling or expressing completely different cell types or tissues and potentially suggesting or prioritizing some type of mechanism in a particular context. So, sorry, So yeah, the idea was so we more or less suggested that this looking at genetic studies through this gene module approach is helpful especially for an interpretability point of view also. So I don't know if anyone has questions. I'm gonna move to a more ongoing and future research now related to this, but if you have questions, interrupt me at any point. So something that we want to do is to improve these latent gene expression representations. I showed you it's clear that the concept of gene modules is key in this approach, of course. I showed you examples like LV246, where we have a very clear signature. However, this is not always the case. An LV could be a technical artifact, could be something that we are not interested at all. It could be capturing that variability. Or sometimes it's very hard to, the RNA-seq metadata is very low quality. So this is sometimes it's very hard to infer the context where the LVX is expressed. We also use models that were built for other purposes, not for integration in genetic studies. We are working on that right now, so they could be suboptimal. We are trying to make the methods that capture these modules more scalable to apply them to larger data sets like Recount-3 or ARCH4. Another limitation is that these are linear only unsupervised methods. So we want to see if we can apply, we're actually working on this, building deep learning models that are also interpretable or applying coefficients that capture nonlinearities in the data. This is something that we published recently. We published a method called cluster match correlation coefficient. So this is a method that can capture non-linearities in the data. There are many methods that are similar, but CCC is faster. So we capture these different gene pairs with nonlinear patterns in GTEx. This is whole blood. And these patterns were missed by linear-only coefficients like Pearson or Spearman. However, they were, like, for example, this pair replicated in tissue-specific gene networks. This pair in particular is very interesting because you have two linear patterns that are replicated here in an independent data set. And we also found some pairs where the nonlinearity is explained by sex differences in expression. For example, these genes UTY and KDM6A are from the Y and X chromosome respectively, and these linear patterns is across male samples, and this flat line here are female samples. And this one that I showed before, these are samples, pre-mortem samples in GTEx, and post-mortem. So the death of the organism has an impact in expression. These two genes are highly correlated in both conditions, but this linearly, but this represents a nonlinear pattern when you take it together. So we want to take the modules that we have, and we don't want to miss these connections that could be important to understand a particular disease. Okay, so that's my presentation. This is my lab. And the one that you can see here is Hao-Yu. He's a software developer. He's trying to implement CCC in a GPU. We have some interesting results that makes it much faster. Mark is working on applying this gene module perspective to understand the different comorbidities in Down syndrome, where we have a triplication of chromosome 21, and we have a hypothesis that this is affecting these gene modules that are associated with these comorbidities. Jill is a PhD student. She's working on trying to disentangle these networks and understand the edges and filter those edges to come up with a more biologically interesting understanding of why those genes are linked together. And Christina is working on a different projects. She's building prediction models for RNA localization. And Prashant, he's a mathematician, also a postdoc as Christina, and building these deep learning methods that hopefully capture these nonlinearities. So thank you so much. That's my presentation. If you have other questions, I will be very happy to answer them. I kept thinking about Chris' question about the gene core and gene peripheral definition. So if you want to say something else, I would be very happy to hear you.
Wonderful. Thank you very much, Dr. Pividori. So yeah, we have about 9 minutes for questioning. I see that Chris has his hand up, so feel free, Chris, to ask your question, and then also Dr. Pividori to expand on Chris's previous question around regulatory genes as well.
Yeah, thank you very much. Great presentation. And thank you for answering my earlier question regarding master regulators. I actually had a separate question. Would you like to elaborate on the master regulators question first before I ask that question?
No. I was gonna ask you if you want to elaborate more on your question. I think we are still understanding this definition of core peripheral genes. Jill, for example, the student that I mentioned before, she's trying to use trans-QTL analysis to convert this essentially gene co-expression network into a refined gene regulatory network. So maybe some of these edges are false positives. They are not there. They are just correlations. So we start with this hypothesis of core and peripheral genes, but maybe we could come up with a more refined list of definitions for genes. That's the only thing. I'm not advancing much in my answer, but it's a very interesting question. But go ahead with your other question, please.
Yeah, my other question, I noticed throughout your presentation that a lot of your methods rely on TWAS data and good quality TWAS data. And I know that for certain conditions, um, there is there are bursts of transient gene expression where we see rapid changes in gene expression for a short period, and in some cases they are reversible, um, meaning that the transient gene expression can occur, then the effect on the phenotype can happen, and then we see a reversion back and the damage has already been done. Um, were there were there efforts made in the TWAS collection and organization to account for transient gene expression, or was it for the specific conditions you were studying that this tends to typically not be relevant?
That's a fantastic question. I think you are helping us to understand how complex this is. My short midterm goal is to incorporate into these networks not only information about how variants change, the abundance of expression in cis, in trans, also how we also want to incorporate alternative splicing and the other molecular mechanisms that I showed. What you're saying is another layer of complexity that I don't know much about, honestly. But yeah, I don't know what else to add here. But I love to learn more about this transient definition that you mentioned and see if it can be incorporated in these approaches.
Thank you. I guess I'll provide maybe like a little bit of like framing for why I'm asking these questions. So, um, a lot of my research in the past has dealt with cancer cells where these cells exhibit some of these kinds of behaviors where for the question I just asked regarding transient gene expression, there's this very well studied phenomena called the epithelial mesenchymal transition, where cells will experience a random, no, not random, a very rapid, quick change in their gene expression that causes them to begin to metastasize. And then many of these genes that do go this transient expression are reverted back to normal levels after this transition has occurred. I guess the previous question regarding master regulators, a similar thing is in cancer, we often observe these master regulators playing an incredibly important role. In particular, I'm thinking of P53 and RB, where there's so many different downstream effects of these genes, and they have very strong and profound implications on phenotype. And typically, the passive approach has actually been to restore function and drug these master regulator genes. I was interested to see if the if your approach captures these relationships.
No, I would say no. I understand from your definition is that, for example, when I think of a gene module or even a refined version of this network, and I think of the edges in the gene, is it correct to say that this transient definition that you mentioned means that the topology of this network changes over time? Is it correct what I'm saying?
Um, I'm not sure what you mean by topology, but I think that if you have these different modules of genes that certain parts of them or even entire modules may be more active at certain periods.
Mm-hmm.
And in the context of transient expression, uh, typically we mean that one section tends to be hyperactive for a very
Mm-hmm.
short period of time before then being switched off or being reverted to very low levels.
Interesting. So, for example, Mark, this postdoc that I mentioned before, is working on Down syndrome and using these approaches to understand why people with Down syndrome is predisposed to particular comorbidities like Alzheimer's disease, for example. So the hypothesis that we have is that, which I think resembles a little bit about these ideas, is that these genes in chromosome 21 that are mostly overexpressed are affecting these networks or modules and having a cascading effect, and at some point reaching these core genes and having an impact on these phenotypes and comorbidities. And that's how our hypothesis, we are still working on that. So yeah, the answer is I don't know if we are capturing that. I would be very interested in see if there is if this idea of gene networks, if these networks are preserved, and as you said, some genes have changed their effect on other genes in time or in different conditions, or if these networks are not preserved at all. That would be interesting to know. But yeah, I wouldn't say that we support that. Yeah, I don't know if you want to add something else.
I guess since there's a last minute, I'll input that in the context of my past research in cancer, it is we observe these transient gene expression changes in tumors where they can be observed in adult patients where it is sometimes easier and more feasible to collect data. And for some of these other syndromes you mentioned, like Down syndrome, perhaps the transient expression occurs much earlier in development, where it is much harder to collect data.
Yeah. Fantastic. Thank you so much, Chris. I'd love to learn more about your research and talk more about this. I saw that Lauren Richard mentioned something interesting about this. I don't know, Lauren, if you want to
Oh, I was
comment more.
yeah, I mean, I was just sort of commenting. I know we're out of time, so wanted to be respectful of that, but basically saying that the gene function isn't necessarily changing in when there's a difference in transient reactivation or expression of a gene in cancer. So I would think that your models would capture the accurate relationship between genes, even that if their expression is altered in pathologic states.
Fantastic. Thank you we run out of time, but thank you for these questions. I'd love to talk more, honestly.